This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 192, recorded on July 19th, 2012. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast about viruses. Joining me today from Western Massachusetts, Alan Dove.
Great to be here.
It's a little cloudy today, isn't it?
Yeah, cooler than the blazing heat we had all week, though, so that's good.
Did you have a massive thunderstorm yesterday?
Yeah, it mostly missed us, but it was just lightning all around and sounded really bad.
Yeah, here at about 4 p.m. it got dark.
I saw some pictures of New York City with this cloud above it, just
amazing,
just hammering the place.
It's currently 24 degrees here. Yeah, it's cloudy. It's okay. We need it to cool off. Also joining us today from North Central Florida, Rich Condit.
Hi, fellas. How you
Hey,
doing?
Rich.
How's the weather down there?
Same as you, 24. We just had a thunderstorm pass through, so it's wet, and it's still overcast. This is all good. We need the rain.
Wow, we've got a lot of rain activity in much of the country, don't we?
Yeah, well, that's all good. We really need it. We've been in a serious drought, so it's good. We need several more feet so I can float my boat. That's what's important.
That's right. Yeah, I think the Midwest has really been severe drought. They're talking about corn crops failing and all that sort of thing.
You have not gone out in your boat yet?
My boat is in dry dock, basically, because the basin that I normally keep it in has been dry because the lake's down 6 feet.
Oh,
Wow.
so you just keep it in this lake?
Yeah, I keep it in a lake.
How long a boat is it?
It's just 18 and a half feet, but it's got a fixed keel, so it draws 2 feet of water.
Neat.
There you go.
Well, we took our boat out a couple of weeks ago.
Are you finally sailing now?
Well, we went once.
Have you got the spinnaker up yet?
No, we didn't get the spinnaker up. It was a really, really windy day.
And
yeah,
you don't want to experiment with that on a windy day.
No.
One of the problems with I think it was during the July 4th week, and there was a lot of power boats. Sailors say the Italian mafia is out. Mm-hmm. That's what they call it. I'm not insulting Italians because I'm Italian. Okay,
right.
And there's so many wakes. The problem was the wind was cutting right across the wakes, and we're trying to go downwind, and you can't really go right over directly 90-degree angle to a wake, and you get smashed all around. So it was very difficult to manipulate.
The perfect day is hard to find.
So we almost turned over.
Capsized.
So the boys are capsized. The boys are worried because we have never capsized, and they're not sure they could get it up, So,
you have to. You
got to go out and just do that.
Go to any small boat. Go to some protected waters. Stand on the rail, grab the mast, and lean. Whatever you have to do to get the boat capsized, and figure out how to get it righted again.
Yeah. So when we launch, we're in a protected cove, so it's perfect.
Now you have it's a little one design racer, right? What type?
It's a laser.
A laser. Piece of cake.
It's a 5-person laser. It's 15 feet. But to launch it, we roll the cart into the water and float it off. So we're standing there. We could capsize it right there. So they're not. They don't have the confidence that they could do it. I said, no problem. You guys can do it. But
yeah, go out where the water is deep enough that you would have to tread water, so you get the real experience. But on a laser, a laser is a bit like a sunfish in this respect. I've brought those back up just by not even lowering the sail, just standing on the dagger board.
Yeah, that's the key. That's the method. You stand on the dagger board, and I'm going to send you a picture of it right now. It's what a lovely boat.
They're fun. They're fast.
It's a little bigger than.
You have a little more cockpit space than a sunfish.
We have. I'm going to put it. Oh, you're on the wrong here. You're on the wrong thing. Oh well.
That's okay. I know what a laser.
Search for Laser Bahia. BAHIA. It's a little bigger than what you're thinking about.
Than the standard.
Yeah.
Oh, yeah, this one. Okay.
So they got some photos with the spinnaker up, but we haven't done that yet.
Cool.
You may have to drop the sails on this one to bring it upright, but the first thing I would try is stand on the...
Is that right? Sometimes you have to drop the sail?
In a larger dinghy, the sail, especially if you get... if you capsize and you have enough time for the sail to sink a little bit.
Yeah, right.
It's a lot of water. Just make sure you release the sheets.
Yeah, right, of course.
So you release, so the sail can actually flop and come back up. And then when you stand on the dagger board, stand close to the hull on the dagger board, because if you stand all the way out, you might break it.
Break it off, yeah. All right, well, that's this week in sailing. Any questions? twiz@twiz.tv. And now we have an all-email show today on TWiV.
Yeah.
And we have a couple of follow-ups before that. The first one is from Colm, who wrote a comment on the TWiV 190 episode. Do you plan to do any promotion of an ASV 2012 hashtag for the meeting in Madison this year? I remember some limited tweeting from Minneapolis last year in between melting into the sidewalk. But I think TWiV would be a great way to promote it ahead of time, along with using it when you tweet about the TWiV episode to be recorded there. After the discomfort with live tweeting the morning sessions last year, it may not be encouraged, but I think ASV could be moving faster in the social media realm. As you said on Twitter, there was a specially designed app for ASM this year, and I know Society for Neuroscience has one too. ASV, as far as I can tell, doesn't even have a Twitter account, and only the barest of Facebooks. With all the focus on H5N1, loved the discussion. This seems like the perfect time to expand ASV's reach on the internet. Maybe I'll apply to run their social media presence once I finish my PhD someday. Because it feels like that when you're working on your PhD. Yeah, the ASV does not have a great social media presence. You know, that society exists just to run this meeting, and they don't do much else. Although I am supposedly on the education committee.
Yeah, I was going to ask if you were still on council.
I'm not on council. I'm on the education committee, and I'm supposed to be bringing them into the social media realm. But I haven't been doing my job.
Well, get on it.
But I did tweet last year from the plenary sessions, and it did raise a little bit of eyebrows. People didn't quite like it, but I'm going to do it anyway this year.
Yeah, I think that kind of blew over.
Yeah,
They'll get used to it.
But you're right, Colm. Anyway, the hashtag is going to be ASV12 for any of you. And by the time you listen to this, ASV will be going anyway, so it doesn't matter. I'll see you there, Colm.
Yeah, and live tweeting from the conference, unless you're having a Gordon conference or something and you've specifically banned journalists or anybody writing about it, then it's a public conference, right?
I think so, yeah. Everybody's there.
If people can write about it the next day, they can write about it while it's happening, right?
I don't see what the issue is.
Yeah.
The next one is a response to an email on which we read on 190, I think, and it was about 175. This was a paper that where they looked at these micro skin patches for immunizing mice, and they used flu as an antigen. You guys remember that? Were you on the show?
Yeah.
And the original email, which I read on 190, we didn't have an answer for it, is why didn't they see any dendritic cells in the lymph nodes? So they saw them in the skin, which was immunized, but they didn't see them in lymph nodes, because normally dendritic cells go from the skin to the lymph node, right?
Right.
So I had emailed Dick Compans, and he actually answered this week. He was the lead author on that study. No sampling of lymph nodes was done in the case of the in vitro cell migration experiments using excised ear tissue, from which we observed the emigration of DCs. For the skin immunization on the dorsal surface of the mouse, we examined the inguinal lymph nodes at intervals post-immunization. We think that several factors may have contributed to our lack of detection of cells containing labeled viral antigen in these lymph nodes. Retention of some of the antigen at the immunization site, problems with insufficient labeling, timing or quantitation, or possibly the processing of antigen. We don't anticipate that any novel mechanism is involved. Additional work is underway using improved procedures for labeling of the antigen, which we hope will resolve this question. Thank you, Richard Compans, for answering. I appreciate it.
Yeah.
Very good. So it's just technology and technique.
But good catch. That was something that should have been, you would expect to see in the paper, and you didn't. And good question.
Good. Rich, why don't you take the next one?
Right. This is an email I got from Yan Zhang, who's a professor at the University of Texas Health Science Center in San Antonio. He just sent me an email after the last show where we talked about recombinant vaccines that recombined and made an infectious virus, and the question came up as to whether anybody's ever sequenced viruses, in particular pox viruses, out of people who'd had vaccine complications or whatever. And he writes, Rich, I was listening to TWiV on my drive to work today. Just want to let you know that there is at least one paper by Chris Upton, David Esteban, and co-workers describing the sequence of a vaccinia virus strain derived from a person who experienced complications from smallpox vaccine. Here's the link. And he links to a paper that's in Virology Journal from 2006. And the patient in question it's a little vague. It's an archived sample of some sort where they had, I guess, clinical material, scabs and stuff from this individual who came to probably Duke University Medical Center, because it's vac virus Duke. Yes, presented to a doctor at Duke University Medical Center. But it's not clear how this woman got infected and why she was experiencing a complication. She had vaccinia necrosum or something like that, which is sort of a spreading and necrotizing lesions. And the virus that she was vaccinated with Dryvax, which is, we now know, it was not a cloned virus preparation. This is the stuff that was used during the smallpox eradication campaign and for years in the U.S. It's not a cloned preparation of virus, so we now know that it's very heterogeneous with respect to viruses. It had, and if you pick 12 different plaques, you'll get 12 different sequences. And out of this woman, they got just one sequence. So, and once again, they're a little vague on exactly where the clinical material came from, whether it was one or multiple lesions, but at least the suggestion is, actually it's interesting that if you vaccinate somebody with something heterogeneous, what grows out looks clonal, as if that person, the complication that they experienced at least, was due to a single strain of virus coming out the other end. But can't say, they have some stuff about genes that are there or missing relative to reference strains, but you can't really say anything about whether this strain actually was responsible in any way for the complications. But at any rate, that is a documentation of sequence of a virus that came out of a person with a complicated smallpox vaccination.
Okay.
Right. There you go.
Very good. I'm glad that he picked it up in his car. Cool.
Yep. Oh, did I finish? Here's the link. Love the show, he says. Yep. Okay.
All right. So let's move into our email, and we'll start with Alan.
Sure. Ian writes, Dear hosts of podcasts, viruses, and more, I've just finished listening to the excellent TWiV 183, and I can't help make a certain observation. In this episode, but also generally, it would seem that the TWiV crew may suffer from a case of bioinformaticsophobia. Guilty. I wouldn't be too concerned, as I expect this affliction is very common. In fact, I have attacks from time to time. Since TWiV is so successful at teaching many of the other aspects of and techniques of virology, why not confront bioinformatics head on? It would be a blast to invite a bioinformatician or computer scientist onto TWiV to talk about some exciting virology-related computational work and really delve into the details of bioinformatics methodology. I've heard awesome explanations of cutting-edge molecular techniques on TWiV, but when difficult bioinformatics gets mentioned in a paper, I'm often left wondering what's going on under the hood. Anyway, keep up the great work, Ian. And he also sent a link, says, I thought this was a cool random example of bioinformatics at the cutting edge of virology. And if you found it on a search for influenza and bioinformatics, and it's Shannon et al. Looking at cross-reactive CD4 T cells against H1N1 swine flu.
They used some bioinformatics to predict epitopes, and then they tested if they actually exist. Yeah, he's right. We do shy away from bioinformatics. None of us have training in it. It's a very new and distinct field, attracting computer people, basically.
Right.
And it's fertile ground for people who are interested in both biology and computers. There's a big need for this.
Yeah.
Huge need.
So we should get someone who could... we had a TWiV a long time ago with Chris Upton, who actually was on the paper that you just talked about, Rich.
Right.
And he talked a little bit about it.
Chris himself is not really trained as a bioinformatician. He's good at organizing people who can do that kind of stuff to put together a website and that kind of stuff. But if you really want somebody who is well versed in whatever is under the hood, we should, look around.
Yeah,
didn't we have? Was it somebody from Ian Lipkin's lab or? We didn't talk specifically about bioinformatics, but I remember somebody on who does a lot of this sort of thing.
So I had a guy from here named Raul Rabadán, who was a bioinformatician.
Yes. And
he's a pure computer guy, absolutely. And he could explain it. He's really good.
Yeah, we need somebody that we can talk to without a modem. Sometimes these guys can be hard to understand.
Very good. I like that. We did have a guy from Ian's lab who uses bioinformatics as well. But Raul is just a computer guy. That's what he does. So, we could do a bioinformatics 101, just the basic stuff. Like great. a Bayesian whatever. Explain what the heck
whatever
it is.
That'd be good. Yeah. I'd like to know.
Yeah, so I was just reading a paper where they used a lot of bioinformatics. And this was in a journal where they published the reviews. And I was looking at the reviews, and one guy said, why didn't you use this tree-forming thing instead of that tree-forming thing? Blah, blah. I didn't know why. I have no clue why you would use one or the other. So we could get... Yeah, that's a great idea.
Yeah, I could use a bioinformatics 101.
Yeah, me too.
Yeah, and of course, it's reflected in the next email from Jim, who says, just listen to TWiV 183 as usual. It was fabulous. I'm a big fan of TWiP and TWIM. I do think it would help to get a participant to interpret the bioinformatics, which seems to be increasingly present in the more recent articles. It's subtly put. Very good. Yes. Was delighted to have DDD present again. And of course, he's gone. 73, Jim.
What's 73, Alan?
Best regards.
Okay.
So this is another Jim. This is not Jim from Virginia. I don't
know
where this one, but he is different because he always signs at 73.
Right.
But that's a ham radio thing, right?
That's a ham radio thing, yes.
Okay. Rich.
Stephen writes, I've been listening a lot to a lot of TWiV, TWIM, and TWiP recently. I'm learning a lot and being reminded of a lot. You touch on insect biology a number of times, but I wonder if you or maybe a listener knows of the Racaniello and Friends of Entomology. I'm thinking someone should create something very like TWiV, wide-ranging but academic, not restricted to agricultural pest management or the global fight against the bugs are icky meme. Fun but information dense. Of course, my imaginary podcast does not exclude the topics covered in the TWiV family. There must be tons of research going on in insect biodiversity, ecology, phylogenetics, genetics, genomics, am I being redundant? Systematics, behavior, e.g. mimicry, species description, biomechanics, e.g. Berkeley IB department, etc., and of course the multi-layered symbioses you folks have mentioned. Interspersed with the history of entomology, Darwin was a beetle collector, and descriptions of the orders and particularly interesting species. I think this could be a wonderful resource with a nearly endless list of potential topics and papers to discuss. Regarding audience, I would venture that the participants in bugguide.net represent a fair number. So Wikipedia link here. Bug Guide had over 809 million hits in 2010. Well, it sounds to me like he's got the whole agenda for a show here.
Yeah.
Sounds to me like Stephen himself is a bug guy. Go for it.
Are you a bug guy, Stephen? And do you want to do this? I'll do it with you. I'll ask dumb questions. I'd love to.
TWiV? I'd love to join you too. I'm fascinated by entomology.
Stephen, I'm putting together a podcast production thingamabob or a company, and I'd love to have a bug podcast in there. So if you want to do it, contact me, and we'll get it going. I'll do all the tech here.
It's like there's more bugs than anything, right?
Yeah. Well, except at the macro level, there are more bugs than anything.
There are more bacteria than anything.
Yeah, okay.
Or maybe viruses, I don't know. Yeah, nice. The next one is another Stephen, huh? It's your turn, Alan. I don't know if it's the same one or different. I can't tell.
I'm guessing maybe it's a different one. But anyway, somebody named Stephen writes, in TWiV 46, you discussed Israeli acute paralysis virus in response to an email. At about 3440 mark in the show, you both chuckled about the idea of putting a radio tracking collar on honeybees. I remembered posting about just such an experiment several years ago on my animal communication site. The photo he's got here shows a radar transponder on a honeybee, and sure enough, it does. It looks pretty cool, actually.
It's amazing.
It's this bee has this giant antenna sticking out of it, and I guess
I wonder if it messes with the bee's ability to fly properly.
It must to some extent, but
it's big. It's like us strapping a rake onto our back.
Yeah.
Wow.
There's another link here to some other pictures that are pretty good.
Then they tracked the bees with radar and followed their flight patterns. It's a very, very cool technique. So he says that was a 2005 Nature paper, and technology has advanced since then. And he hits us with a couple more links. So yeah, you can potentially track this, use microchips or what have you, and
yeah. I guess we were wrong to chuckle.
Well, it's still pretty funny.
We can chuckle anyway. It's still pretty funny.
It doesn't deny the humor in having the giant antenna sticking out of the back of a bee.
Right.
Reminds me of the Far Side cartoon with the bear coming home to his wife in the cave, and he's got a big target on him and a hypodermic syringe hanging out of him and a radio collar, and she says to him, "You'd better have a pretty good story."
That's great. Yeah, they're great. They're just great. The next one is from Robin, who sends a link to a PhD comic called The Evolution of Intellectual Freedom. This is just great.
Yes.
We start before grad school. I'm going to research whatever I want. Grad student, I'm going to research whatever my professor wants. Assistant professor, I'm going to research whatever my tenure committee wants. Tenured professor, I'm going to research whatever my grant committee wants. And finally, emeritus professor, I'm going to research whatever. And then RIP is research in peace. So what stage are you at, Rich?
I'm pretty close to RIP here.
No, you're not close to RIP. I think you're a tenured professor.
Actually, I'm really lucky here because, I got mine. I'm somewhere in between whatever my grant committee wants and the whatever. I guess this guy can't remember what he's supposed to be researching. He's that old.
Right.
I'm in the I can research whatever I want phase.
Yeah, it's good.
It's a blessing. So
nice
it is it is true that people talk about academics. You can do anything you want, and that's not entirely true. You have to convince somebody to give you money for it.
Right.
Yeah, once you have money, you have 5 years, you can pretty much do what you want. But if it doesn't work out, then you're going to have some issues, right?
You can do what you want as long as it's going to show results in that time frame.
Rich.
Let's see here. Stephen writes.
Another Stephen.
Another Stephen? A quick email response to TWiV 181 in which Kathy used APOD, that's Astronomy Picture of the Day, as her pick of the week, which I now have as my homepage on my browser. Rich asked how he could get this as his desktop wallpaper. I have it on my Windows 7 desktop, but not as wallpaper. As seen in the attached images, I have APOD at the bottom of a screen in the panel and shown progressively larger if one wanted to have it done. A program called Rainmeter, he gives a link, can display a number of skins/themes. This particular theme, called Omnimo, gives another link, is designed to look like Windows Phone 7 or Windows 8. As ever, thanks for keeping me informed and entertained. So I looked at these. These are basically customized desktops for PCs. And so there's a couple of different ones. Well, I guess there's an overall shell and then a more custom one made out of the same thing, this Omnio. And they look pretty cool. And then he sent us a picture of his. I noticed that on his desktop he's got the 2 Reforming Science articles that we discussed. So he's
right there. Look at that. Cool.
Yeah.
Hey, good eye.
So, great. That's it's like turn your desktop into an iPhone or something similar. Cool.
All right, Alan.
Let's see. So that was that one. And then Lance sent by Twitter. This letter is just a link because, of course, it was sent by Twitter, but I think we're probably going to spend some time talking about this. This is a measure that the government in the UK is proposing, I guess, that's going to require scientific research funded by the government to be made immediately available, open access, starting in 2014. And that's the gist of it.
It's amazing.
As I understand it.
Yeah.
Yeah.
Amazing.
The journals must be screaming, but maybe they don't
Well,
have
actually, they're trying to arrange it here so that the publishers won't necessarily get thrown out of business, okay? Because they're shifting the burden of paying for this from the libraries to pay charges to the investigators.
Yeah.
Right.
So my guess is, I don't know if the investigators are going to scream about that because, of course, everybody's, it's going to cost, they estimate, £2,000 to publish a paper. That's a lot of money.
We're paying page charges already, right?
For a lot of journals, some not.
I mean, the open access PLOS journals, they all, that's their model, right?
Yeah. Yes, right.
Which account do you want the money taken out of is really the center of this question.
Yeah.
Right? You're paying for it. There's no way around paying for it. You're either paying for it by your institution's paying for it out of a subscription fee, in which case you're ultimately paying for it out of a grant somehow, or you're paying a page fee for the research that you publish, in which case you're paying for it out of a grant.
At least this lets the public get to it, right?
Yeah,
exactly.
I mean, they are still rationale. They're paying for it through the grants, through their taxes, but at least they don't have to subscribe to every journal or pay $35 for a reprint.
Right.
Which I think is good. The real question is, will this ever happen in the U.S.?
Well, there's already this NIH policy for opening up the research. What is it, 6 months after?
6 months, I think.
Yeah,
right.
That's right.
So that's PubMed Central. And that has been around for a while and appears to be working okay. I mean, somebody can write in and tell me if there's some horrific failure of it that I haven't come across, but that's there, and people have made noises about, well, when are you going to do away with this 6-month window?
Right.
So really, this is not some completely radical new idea that the Brits have proposed. They're just taking the same approach that the Americans have taken, and they're doing away with the 6-month window.
The 6 months is to give the journals some income, right?
Some exclusivity and but the rationale for that from from the NIH has been, you need transition time for journals to adapt to this new landscape, and I don't know how long you wait for that to happen.
Yeah.
So I suspect this is the type of proposal that's being put out there with the idea that it'll be negotiated.
Right.
Yeah, this is not without discussion, this UK proposition. A lot of people don't like the idea that you're shifting research dollars to, the publishing. So who knows?
But, it's the whole thing is moving this way. It's. I think the whole immediate open access thing is becoming inevitable. There's such a movement that way, and there's so many of these journals, open access journals, that are successful.
Yeah, and a lot of the subscription journals are now going to a model where they give the option.
Mm-hmm.
So you can choose to pay a pay charge, and they'll publish your paper open access. Or you can not pay the pay charge, and they'll publish it behind a paywall. If you had a policy like this and grant money that accounted for it, then I guess the British researchers would have to choose to pay the pay charge and get it published open access. So that kind of business model can serve as a transitional point.
I mean, all of this is to keep the journals in business, right? If you envision a system without journals, I mean, they could still publish, news and views articles, but without publishing the primary literature. Let's say the governments just took care of peer review and publishing the journal articles. Would that work?
I don't really want the government doing it, depending on how you define the government.
Yeah,
I think
I'm not sure I trust that approach, because the government is subject to the whims of budgeting decisions. I mean, we're watching right now, there's this political football being kicked around in the U.S. about what type of NIH funding is going to happen.
Yeah.
And there are some programs that are unpopular with some politicians, and so they're trying to cut them. And that's what happens. What happens if somebody decides that they don't want to keep supporting this scientific publishing thing?
Yeah, I think you're right about the government, but as we have said before, the journals have issues. There are conflict issues as well. It's not perfect either way.
Right. Either way, but the journals exist already. The government system is something you'd have to put in.
Brand new. Yeah, but would you say that in 50 years there's going to be something that we don't even know about today?
Quite possibly.
Or is it going to look? Is it going to look just like it is, just slightly different flavor?
I'm not sure. There's been a lot of talk about whether this whole idea of the paper will persist.
Yeah.
You right now you amass a certain amount of work that qualitatively looks to you like a paper, and then you take it to publication and it goes through peer review, and that's a unit of research. But why?
Yeah, right.
If the lab notebooks were open, then you wouldn't have that kind of publication. You would just publish your data.
And who's to say that your interpretation is the right one? Maybe someone else would take the data and reach some other conclusion, right?
Right, and why spend the time to write up an introduction that's going to be redundant with all the other introductions that have been written in your field, and a discussion section that's your interpretation? You could certainly put those things on your website, but if you just put the data online,
it's training for graduate students.
Interesting.
Exactly. And there's this, when you get into these discussions, a big part of the problem is we've built our whole infrastructure around the assumption that this is how science is going to be produced.
Yeah, right.
And there are a lot of advantages to doing it that way. A paper or something of that sort really focuses your mind on a series of experiments as a thematically consistent set of questions, and you put it together in that way. And if you're just publishing data, well, why do this experiment and not that one?
Well, I think we ought to revisit this. I have the editor of Retrovirology. I met recently, and he'll come back on. He'll come on TWiV, and we'll talk about it again. We'll get his perspective.
Online open access journal.
Yeah. And we'll see what he has to say.
Cool.
It's really something. It's not going to go away, and there are no answers, clearly. All right, next up is from Matt Freeman, who sends us a handy temperature chart for TWiV, which is absolutely fabulous.
I was going to use exactly the same word. I immediately posted this on my Facebook page. It's wonderful.
So we got Fahrenheit, there's a line, 0 degrees really cold outside, 100 degrees really hot outside. Then versus Celsius, 0 degrees fairly cold outside, 100 degrees dead. Versus Kelvin, 0 degrees dead, 100 degrees dead. That's great. You know, when I first got it, I thought, oh, another kind of scale thing. It's great. And then you realize it's really funny. Thank you. Thank you, Matt. All right, let's skip this one from Angela. We'll save this for ASV. Move to Ricardo. Is it your turn, Alan?
I just did the link.
The link. Okay, so it is Rich's turn.
Okay. Hello, TWiV tertulia.
Remind us, what's tertulia?
Tertulia is defined as Wikipedia by it's a Spanish Italian derivation, is a social gathering with literary or artistic overtones, especially in Iberia or Latin America. On TWiV 174, a Google engineer asked for a test that would help him figure out the respiratory infections in his house. I just found out a test named XTAG RVP-V1 and XTAG RVP Fast that are able to qualitatively identify several respiratory viruses. Probably expensive, but it is information. Congratulations on your growing numbers, and keep up the wonderful work. And I looked up these things, and RVP is Respiratory Viral Panel. These are tests that are produced by Luminex. Luminex is basically a flow cytometry-based analysis company, and they've got a nice, I put a link here. If you browse around their system, you can find a video that describes how this works. And basically, you take a nasal swab and put it in some sort of mix that in a tube that's obviously part of a kit that makes cDNA and amplifies that, and then does, using candidate virus specific primers, does a reverse transcription in the presence of, I guess, a fluorescent label, so that now you have a labeled a fluorescently labeled DNA that is amplified with a specific primer specific for a given virus. And that primer has on it a tag that will hybridize specifically to a bead that's got a complementary nucleic acid on it, and it's specific to that virus, and the bead has a specific color. So each candidate virus has a specifically colored bead, and it will grab onto a labeled nucleic acid if it has been amplified. Then you stick all this into a machine that's like a flow cytometer that looks at each individual bead and says, yellow colored bead, that's flu. Does it have a tag on it? Does it have a labeled tag on it? Yes or no? And that tells you whether or not that sample had flu in it.
Right.
This is not something you're going to do at home.
Well, depending on how long this particular Google engineer has been with the company and what stock options he has,
right.
Buying $50,000 luminometer type setup might not be. I don't know what those things actually cost, but that's just a guesstimate.
So I found the technology fascinating, and the video describing how it's done is really, really nicely produced, and it gives you an idea of where the state of, I don't know if this is state of the art, but it's certainly a state of the art, and shows you what people are going to be capable of doing. I really thought this was cool.
Yeah, and this particular thing has apparently been FDA approved as a clinical diagnostic test.
Mm-hmm.
So it's probably in use now in some clinical labs.
Yeah, So I suppose, a doctor could take a sample and put it in a tube with some sample collection buffer in it and probably send it off to this company, and they would do it.
Well, or if you had probably you'd send it to a diagnostic lab.
Yeah.
That's the kind of facility that would actually buy this gear, and they would run enough tests to make it pay.
Okay.
And that could be really useful. You could test somebody and get an immediate feedback or very fast feedback on whether they have a bad rhinovirus infection, or if it's the flu, or if it's pneumonia, and maybe reduce inappropriate use of antibiotics and antivirals that way.
So this particular kit, I forget how many viruses it.
8, is it?
Something like that. But I'm sure that this will get.
8 viruses and subtypes.
Okay.
Flu A subtype, H1, H3, flu B, RSV, human metapneumovirus, rhinovirus, and adenovirus.
Cool. There you go.
Hey, can we use tertulia in the title?
Probably.
Oh, yeah.
How about email tertulia? or
viral tertulia.
Viral tertulia. All right, I like tertulia. I want to use it.
Yeah, that's a good word.
All right, I think you're next, Alan.
Probably. So, Ricardo. Wait, is this?
Yeah, it's the same Ricardo.
Okay. I guess it's not Stephen. Ricardo writes, "Hello, Vincent and Alan and Dixon and Rich. First off, I'm sorry for this email because it is a request. No problem. I have already written to you about this, but I'm really struggling with it. I can't find a source of information or help to prepare laboratory classes for undergrad virology students. Our lab is we're teaching a lab, and the coolest thing I can do is to find plaques with bacteriophages, and that only takes 2 90-minute classes out of 18. Can you help me here?" And Ricardo is a professor of microbiology in Portugal.
Hey, last week we talked a little bit about this for medical students. So I suggested doing a neutralization assay for polio.
Right.
But why don't you go out and get some dirt and have them isolate their own phage?
Yeah.
Like they do at Graham Hatfield's lab, remember? We had him on TWiV.
Yeah.
UPitt. Isolate a phage. You don't have to sequence it like Graham does, but they could plaque it out, and they might find that cool. That could take a couple of labs, And
I looked around in the Cold Spring Harbor library, basically, to see if I could find some sort of a lab manual, because they used to have lab manuals to do microbiology one sort or another, but I could not find anything there that was satisfactory.
Well, if this is an undergrad class, often undergrad labs are done with well-characterized outcomes. I'm not saying that's necessarily the best way, but it does at least guarantee that you'll get a result. And it occurs to me there's an enormous body of research that was done a number of years ago on phage, and that was extremely important in our understanding of molecular biology. So you could set up and do the classic experiments on phage lambda and M13 and just repeat a lot of the pioneering work with pretty much the same tools that were available at the time. Kind of like what's done in a lot of undergrad and high school labs with fruit flies, looking at eye color.
Yeah, a one-step growth experiment you can do.
Yeah.
I'm thinking, as a matter of fact, you could probably, T7 is so easy to work with. You could probably get, even now from Bill Studier, a collection of T7 amber mutants and appropriate hosts, and at least show what a conditionally lethal mutant looks like. You could probably even do a complementation or a recombination experiment.
Right. You probably can't do any of the radiolabeling studies.
No.
Although you might be able to substitute fluorescent probes for some of those.
Could be.
If they're non-toxic enough. And it is college, it's undergrad, it's not high school, so you don't have to be completely paranoid about the chemical safety.
Man, when the genetics course I had in undergraduate, now this wasn't phage, this was bacterial genetics, basically. It had one small phage component. But I think that this had as much to do with me taking the path that I did as anything else. It was a 10-week course, and I forget, there were probably 2 4-hour labs a week. And in the first 3 weeks, we learned everything you needed to know to do bacterial genetics, basically. We had I can even remember some of the names. We had a female E. coli called AB1157 that had everything possible wrong with it that you could select for. You know, a whole bunch of nutritional markers, drug markers, etc. And we had several different HFRs, and we learned how to do matings on a plate, you know, using streaks, and map the HFRs. We learned how to do an interrupted mating, and that involved growing up a stock of phage T6 and using T6 to interrupt the mating. And then on week 3, the professor, Rick Davern, since deceased, unfortunately, he was brilliant, did a mutagenesis of the coli and showed us how to replica plate and select for temperature-sensitive mutants. And we picked temperature-sensitive mutants, and then we had 7 weeks to characterize the mutants that we got. And his expertise was in DNA replication, and anybody who got a DNA replication mutant got a case of champagne. But I'll tell you, in that 10 weeks, and we were undergrads, and I don't think I fully understood everything I was doing at the time. I got most of it. It was really intense, and it was just so good. Man, it was great. It would take a while to reconstruct that, and I'm sure it's not written down anywhere, but it was just absolutely brilliant. Now, it's not virology, it's molecular genetics, but boy, was it good.
That's cool.
Yeah, the old stuff was great.
Dave Botstein, I think, used to run a lab where he used T5 and did the same kind of thing, where he had a mutagenized stock of T5 and gave it to the students. And, amber mutants would be the best to do in this case. That is, they're host range mutants that have amber mutations. And basically, the students selected amber mutations out of this stock of phage, and then I think as a class sort of did complementation analysis and recombination analysis to map them, and maybe some limited phenotypic analysis as well. So that's another approach that can be done.
Yeah.
Hey, listeners, you got ideas for Ricardo? Send them in. I'm sure there are people out there who have had virus labs that they liked, so let us know. Collectively.
For undergraduates, phage
is great.
Phage is great because it's quick, it's cheap, and it's non-toxic.
But he's got 18 classes, so they can do a virus experiment.
Could
use Sabin, and I would say look for neutralizing antibodies. You got to get somebody to bleed everyone though, and they may not like that because they're not medical students. They don't want to stick each other. All right, the next one's from Marissa, who writes, "Hi, I love TWiV. I learned about it from my virology professor and always listen to it in my car now and at the gym. I was wondering if you guys would consider making an iPhone app so we can listen to it on the road without having to plan it, kind of like the Science Friday one. And maybe a Facebook page, too. Get people interested in science. I'm the only science major in my family of political science buffs, and I am always wanting to get my dad into listening to these. He listens to NPR since forever, but now NPR has an iPhone app, which makes things really easy. Just some thoughts. Plus, I really love your podcast on oncolytic viral therapy. I recently learned about RNAi therapy in regards to diseases this summer and thought that was a very interesting subject as well. Thanks for all the podcasts, and you guys are really funny, especially when you guys first got your iPhone. I cracked up. I recently got one too. Ha ha. Keep up the great work." This is just a stream of consciousness, Marissa.
Yes.
Marissa. That's great.
Nothing wrong with that.
We got a couple of apps you can use. We got the microbeworld.org iPhone app, or you could go to Stitcher Radio. They have an app. There are actually a lot of podcasting apps now where you can download on the run and listen. So
what's the advantage to doing this with an app when podcasting is a built-in feature on the iPhone?
Apple has just separated the podcast player from iTunes, from the music player. There's now a separate app for podcasts.
Really?
Yeah. So if you previously played, if you're keeping your iOS up to date, if you previously played podcasts using the music app, they are no longer there. They have separated out into a separate app called Podcasts.
Oh my God. Well, wait. I listen to it on my computer. I guess that's the problem. iTunes on my computer still has my podcasts in it.
So the OS 10 is still merged, but I think in iOS they're trying to separate them.
Oh.
I don't know why, just to make not such a huge app. I don't know. I don't know what the logic is. But now there's a plethora of podcast players in addition to that have cropped up. I use one called Downcast. There's another one called Instacast. And these all allow you to download the podcasts without having to sync with iTunes, for example. You can do it on the run over 3G. So check those out, Marissa. There are plenty of them. All right. Alan is next.
Oh, sorry. I'm just staring at my iPhone. I still seem to have podcasts in there. I probably have to update. Anyway... Charles writes, might be nice at some point to have a renewed discussion on the incurable wound revisited with the possibilities of rabies virus therapy. I believe Doctors Willoughby and Glasser could be great candidates for an interesting chat on same. Also, with the advent of World Rabies Day on 28 September, would be ideal to have a mention of a rabies topic that week before, if possible. And he sends along a link to an article from the Statesman newspaper in Austin, Texas. And this is actually a really, I think, a pretty well-written article.
Yes, I really enjoyed this.
On this CDC case, the Texas Wild Child case, as it's sometimes called, of the girl who showed up at a hospital in Texas and apparently had an actual symptomatic case of rabies and got better.
So the author, one of the people quoted in this article is the author of the email.
Right.
Charles Ruprecht is a scientist at CDC, apparently. And Glazer is also quoted in this article, who he suggested for. So,
and this incurable wound apparently is a New Yorker article from some time ago, where it was the finding that bats are a major source of rabies in the U.S. was discussed.
Okay.
So basically he's saying, let's go back and have a look at this stuff again. So World Rabies Day, September 28th, huh?
Is that right?
That's what he says.
You would know.
I have to put that on my calendar.
We should, maybe we could do something before that, as he suggests.
Yeah.
Okay. Rich.
That's coming up rapidly.
I'm busy here putting World Rabies Day on my calendar. That's the kind of thing you don't want to miss. Varun writes, hello, microbiology crew. As always, the TWiV, TWIP, and TWIM has kept itself on the top of my priority list. And I must say, keep it up. And all I can say is it has kept getting better and better. I have a couple of questions and a few suggestions. My first question is there any chemical that attacks the geometry of a virus? I couldn't find any, but did find that the viral geometry, once disrupted, will cause interruptions in assembly of at least most of the viral machinery. Please enlighten this area. My second question is, could you elaborate about dead box proteins? This group of proteins has really been difficult to digest intellectually. We can come back to all these one at a time. I have a suggestion. Since you interview various scientists on the show, which has really given all the audience and great peek into current science, it would be fantastic if you could get Stanley Prusner or Erwin W. Sherman on this show to talk about their current research. Also, I request you to have occasional Virology 101. I feel it has now reached near extinction. I know.
That hurts.
Yeah, well, but he's right.
He's got a point.
I also have a listener pick of the week, if you would like. If you consider Matt Ridley's Genome as a masterpiece, which was picked previously in the show, then I guess his other book, Agile Gene, is a next step to masterpiece. That's my pick. Once again, thanks for this highly entertaining program. So let's see. Chemical that attacks the geometry of the virus. Well, there's some antivirals that you know more about than I do, Vincent. The picornavirus antivirals that fit in the crypt or whatever.
Wind compounds?
Yeah.
Yeah, they fit in a little hydrophobic tunnel in the beneath the receptor binding site, and they interfere with uncoding typically. So yeah, that's a capsid-specific antiviral. I guess that's what he means by the geometry, right?
That's kind of that's what it means to me.
Yeah, disrupt. You have all those nice symmetrical interactions. You could probably disrupt them all. Yeah, with a compound.
He's talking about interruptions in assembly. I can't think of any specific compounds right off the bat. But actually, there's an anti-vaccinia compound, but it doesn't really attack the viral geometry. It attacks a protein involved in virus assembly and an assembly of the form of the virus that causes spread. But it's not really utilizing a virus geometry. I'm vaguely thinking, are there any HIV compounds that do this?
That affect assembly.
Right.
The protease
inhibitors.
Yeah, the protease inhibitors would affect maturation, right?
But that's not really attacking the virus geometry.
Not geometry. I don't know of any. I mean, I'm thinking, you block all the subunits from assembling in the infected cells. Mm-hmm. I think that would be a tough one to do because these interactions are pretty strong.
Mm-hmm. But good idea, since what you want to do with a drug is target something that is unique to the virus.
Sure,
right?
That is a good idea. Yeah.
Let me see. Could you elaborate about dead box proteins? Well, I confess, although I know a thing or two about this, I had to go to Wiki for my summary on this. Basically, the dead box, as with many other sort of monikers, refers to an amino acid sequence, DEAD, that is common to these proteins. And typically, they are proteins that bind to RNA and do something, typically involving energy, so that's ATP hydrolysis, and the dead box in particular is part of the active site of the protein that is responsible for ATP hydrolysis. And a lot of these are helicases. I think helicase is an unfortunate word. In most people's mind, helicase means formally something that unwinds double-stranded DNA, but I think, in fact, a lot of proteins that have helicase motifs in them, including these dead box proteins, are more, I would say, motors. They're motor proteins. They're things that bind to nucleic acids and move along relative to the nucleic acid in an energy-dependent fashion, and that can have lots of consequences. Okay, it can unwind nucleic acids, or it can bump into other proteins and knock them off. It can remodel proteins on nucleic acids, and etc. So basically, these are energy-dependent RNA binding movement proteins that have to do with RNA metabolism, and it's like particularly mRNA splicing and initiation of translation. That's about the best I can do.
It's good. The first one discovered was EIF4A, which is a translation initiation protein. It's a helicase that unwinds secondary structure in the 5' untranslated regions of messenger RNAs so that ribosomes can move through it. We show that in my translation lectures.
And can we get Stanley Prusner or Erwin Sherwin on Sherman?
I don't know. Stan would be great. I don't know Sherman. Yeah. Stan Prusner would be marvelous, but he's a busy guy.
Yeah.
Usually most people say yes.
Erwin Sherman's into malaria parasites.
Yeah, Prusner could be for number 300,
Yeah.
because he's a Nobel laureate.
Yeah, he's a Prion guy.
Yeah.
Sounds like Sherwin might be good for TWiP.
He would be, yeah.
Maybe Dixon knows him.
I don't know. I can't ask Dixon anything right now.
No?
He's out west fishing still till August 1st.
Does he think he's retired or something?
Yeah, really. Next one is from Chris, who writes, "Hi, TWiV team plus."
We skipped the bit about Virology 101, didn't we?
Yeah, we're trying to forget about that.
Yeah, we have to do it. In fact,
we should do that.
Just stick it on the schedule, you know?
Yeah.
All right.
Stick several on. As a
Wanna
matter
do,
of fact, you ought to go ahead in the schedule and for, once every month or 5 or 6 weeks, just block out one and make us do it, right?
Yeah, I did that once, and then I canceled it.
Yeah.
Because I wanted to do some other paper that came up that was really cool.
Yeah, because cool stuff keeps coming up, and then also we've got this huge backlog of email and guests coming up.
You want to do one in August? Should we do a Virology 101 in August?
Sure.
All right, I'll put it on the schedule. Chris writes, "Hi, TWiV team and audience. I enjoyed your discussion of polydnaviruses and Michael Strand's work. At one point in the discussion, someone, I can't remember who now, asks how such a complicated relationship could have evolved in a non-integrating virus-host relationship. I think this is a very interesting question and wanted to point out some interesting examples of wasp virus commensalism, which it turns out seems to be quite common. While the polydnaviruses are certainly the extreme example, barely viruses at all anymore, really, there are several examples of other relationships where the virus maintains a lot more independence. There are probably many more examples of this than I am aware of. This is not what I work on, but the first example that comes to mind should be of unusual interest to Rich if he is not already aware of it. A colleague of his at the University of Florida, Pauline Lawrence, Department of Entomology, discovered an entomopox virus in the venom of the parasitoid wasp Diacasminorpha longicaudata.
Pretty good, Vincent.
I don't know. Unless I'm mistaken, it is required for wasp development. Appears to suppress the immune response of the host, fruit fly Anastropha suspensa. Unlike the polydnaviruses, this is a legit virus, replicates to high titer in the fruit fly, and contains all its own genes. It seems that the wasp acquires the virus during development in the fruit fly, presumably by consuming infected hemolymph, insect blood. Adult male wasps are positive for the virus too, but only at very low titer in all body segments. How the female concentrates the virus into her poison gland remains a mystery. The virus appears to be perfectly self-sufficient in the fruit fly. You can passage it in fruit fly larvae, but it seems to lack any ability to transmit from one to the next without the wasp. Super cool. Maybe a virus on its way to integration. Unless I'm mistaken, Pauline has discovered additional viruses in the venom of the same wasp, a rhabdovirus, and maybe some more. As molecular biology tools become increasingly available to the entomology community, I expect that we will see a lot of these types of symbiotic relationships in the venoms of Hymenoptera, wasps, ants, bees. It would be interesting to see if it is only the solitary parasitoids, or if social Hymenoptera also maintain them. The presence of this sort of relationship across such unrelated virus types suggests that there is actually a very large ecological niche for viruses in this capacity, so maybe it is not so surprising that we see the polydnavirus story emerging the way it is. Thought you might think this was interesting. My apologies if you are already familiar. As I said, this is not what I work on, so my apologies in advance to important work in the field, which I've undoubtedly failed to mention. I believe Pauline is mostly retired now, but I'm sure she would be happy to discuss it with you, Rich, if you offered her some satchels. This guy is an insider.
Sounds like it to me.
Thanks for all the work you do. TWiV is undoubtedly the best podcast out there and has made me a much better graduate student. Must have used at least 5 things I learned from TWiV in my qualifying exams. TWiV bump. Do you know,
Pauline? Yes, I know Pauline. She is retired now, as far as I can tell, completely retired. I actually, quite a few years ago, served on a couple of her graduate student committees, but I haven't really kept up with her work. I was aware of this pox virus of insects. It's not a typical entomopox virus. As a matter of fact, I'm not sure that this virus even shows up in the ICTV. Pauline, to my knowledge, is the only one who has ever really worked on it, and I was not aware of this fancy relationship with venom and parasitized hosts. That's very interesting.
Yeah.
So cool.
A pox on your wasp.
So she doesn't show up anymore to work, right?
I guess not. Well, she's in the entomology department. That's across campus. Okay, I had to ride my bike over there for committee meetings and stuff. So it's not we don't interact with those people as much as we could.
Those people.
Those people.
Nice.
Sorry.
Is that like you people? Yeah.
There's a virology club here at University of Florida that is run by another professor in the entomology department, Jim Maruniak. That's really a really nice undergraduate club. He does a lot of good stuff for them. So.
It's a great story. We'd love to talk about it sometime, right?
Yeah.
All right, Alan's up.
Sure. Luke writes, Dear TWiV crew, you guys occasionally talk about the flaws of the publication and grant review process, so I was wondering what your thoughts were on the recent commentary in Nature regarding an investigation done by Amgen. They tried to reproduce the results of 53 landmark papers in cancer research and were only able to do so for 6, even after contacting the labs where the studies were done, as well as in some cases using their reagents or even physically conducting the experiments in the original labs. Although, as you have mentioned, the severity of this problem is different for different fields, cancer research, due to its clinical applications being perhaps one of the worst ones, my guess would be that it exists to some degree everywhere. Ideally, as with the XMRV story, misleading results are resolved, but the Nature commentary shows that this is not always the case. And while in many cases such a study could cause some dead ends in the lab, other cases exist where misleading results make their way into clinical trials, large sums of money are wasted, and patients receive treatments that do not work. I was wondering what your thoughts were on this problem in general, and if you had any guesses as to how prevalent it is in virology specifically. You've remarked on supplemental data, especially with Science and Nature, which would like to squeeze in as many articles as possible. The supplement has become a way to have very short main bodies of articles with frequent references to supplemental figures that make the papers unreadable. However, having the supplemental information means that you can publish all the data that is generated, which would allow for others to see any existing inconsistencies and more accurately judge how reliable the study is. As Bagley and Ellis remark, this would mean the stories would become less perfect, but most people who work in biological sciences already know that biology is sloppy and rarely as clean-cut as we would like it to be. Any thoughts on how the pressure for complete, perfect stories could be lifted given the existing competition for grants? As always, thanks a lot for your efforts to make science more accessible to people outside your field. The casual conversational style that these podcasts have makes them a pleasure to listen to after a long day of focusing on small technical details in dryly written formal text.
It could be science, right?
Yeah.
This is sort of what we just talked about before. Right?
I think we also discussed this investigation done by Amgen in a previous episode. This is is familiar to me. Yeah,
I looked up for. I couldn't find Luke, so it must have been a different listener. But I think you're right. We did discuss it, but I don't know when.
Yeah, and this is something that certainly, whether it was with this paper or other issues, we've touched on this reproducibility problem and its unevenness across fields. And yeah, especially in a clinical research area, there's an enormous amount of complexity, and a lot of these studies will look at a bazillion variables and then come to some conclusion, and somebody else will look at a different bazillion variables and come to some. So it's good that people are in some cases going back and trying to really duplicate the original work. But it may just be that some of these experimental designs had so much noise in them that the signal we thought was there isn't really.
You know where I ran across this recently? I was looking back at one of the articles that we discussed last week about the investigator who runs a 24/7 laboratory.
Right.
Nassim pointed out to me that there was a long and interesting bunch of comments at the end of that article. Mm-hmm. And some of those comments were from people working in industry who said, no wonder we can't reproduce any of the preclinical results. It's because all of these people are, up all night trying to get their careers together in a very competitive environment. It comes back to the thing on reforming science that we talked about some time ago.
Yeah, I agree. I thought of that when I was reading this as well. It's the science has to be reformed because there's too much pressure and you get a lot of this stuff. We talked a little bit about open science before, putting all your data online.
Right.
And that's sort of the supplemental data is one step towards doing that. Yeah.
Completely different publication model. I hadn't thought of that. It's an interesting idea.
I don't know in virology. I think that in virology, most studies are never looked at or never no one tries to reproduce most work in virology, as far as I can tell. Major stuff that has clinical implications, perhaps, or. But my feeling is that most of the stuff we have published, for example, no one else has tried to reproduce or bring forward in any way. Everyone has their own story, and they pretty much focus at that, with few exceptions. And then,
in a lot of cases, you get a sort of a de facto replication when somebody extends the work.
I suppose, yeah.
Because if A weren't true, then B wouldn't have worked.
True, and
so
there are certainly many areas where that happens. And I'm thinking of the whole viral transformation story that went from Rouse all the way to oncogenes and signal pathways, and it all made perfect sense, right? So that's an example of replication. I mean, along the way, there are things that people can't reproduce or they can't figure out, but if it's an important problem like transformation, it gets sorted out.
Yeah, and the reproducibility is maybe not the best term. It's the one that we use, but it seldom makes sense to go into the lab and look at somebody's materials and methods section from a published paper and try to duplicate that exact experiment. What you're going to do is say, hey, wow, they found that. So if we look here, we ought to be able to design this other experiment. And so you come at it from a different angle, which I think is actually a better way to confirm that you're looking at reality, because if the elephant is the same shape from two sides, then it's probably the same shape. Whereas the reproduction, you may just be reproducing an artifact. But that only works in fields where you have, in virology, we're often looking at 3 or 4 orders of magnitude difference between the experiment and the control, and it's not really ambiguous what happened.
Boy, I sure agree with the examining the same problem from 2 different angles. I like that much better than trying to do exactly the same experiment again. Though I must say that for my PhD thesis, I wound up finally trying to reproduce the published work that it was based on about 3 years into my project, and found that the published stuff was not right. And I wish I had tried to reproduce it from the get-go. So if you're doing something that is taking off from something else, it does pay to do something early on to make sure that the phenomenon that you're investigating is really valid.
Sure.
Some kind of a spot check,
Yeah.
I mean, but just given all this, science has done amazing things. We've learned a hell of a lot.
It's not perfect.
Right. It's not perfect, but in the end, really good stuff happens.
So,
I think this does touch on a very important point, that just because we've got a lot of work that does come out reproducible, and in many fields it may even be the majority, that doesn't mean that every experiment is well designed.
Yeah, of course.
The phrase that we've used before is self-correcting. It's not necessarily reproducibility, but self-correcting that it's important. And it's too bad that these companies have to sift through, have to do so much correction, put it that way.
Well,
we ought to be better at getting it right to start with.
If you want to put drugs in people, you have to go from the preclinical data and make sure it's right?
Yeah,
so that's
absolutely.
a special case, yeah. Rich, you're next.
Spencer writes, just as a follow-up, because there were so many groans about Netter's cadaver, this was a...
Remember this?
This was I guess a letter that we read
from him. Yeah.
Oh, it was from Spencer.
Yeah.
Saying that he worked on this cadaver that he knew. It's common for the intended anonymity for cadavers to not work out. In Dr. Netter's case, I recall that he specifically wanted the anatomy class to know that it was his body. When I did gross anatomy, my cadaver was still wearing his hospital wristband. It's a very interesting phenomenon as the first year of medical school progresses. Medical students who spend inordinate amounts of time in the anatomy lab go from being squeamish to being very nonchalant around dead bodies.
That's the idea, right?
I would imagine, yeah. Again, thanks and keep it coming. I especially like the revisit of basic parasitology with Dixon. I hope you don't run out of parasites of minor medical importance anytime soon. There's plenty of critters out there.
I don't think there's much danger of that.
No.
Seems a matter of whether Dixon wants to talk about them or not.
Yes, or whether he's available.
Yes. Am I next?
I think it's yours, Vincent.
Lark writes, "Hello, TWiVians. I read in the BBC's April-June Focus on Africa magazine an article by freelance journalist Alice Klein about health issues in Zambia. Klein notes that medical professionals in Africa are accusing NGOs of bending to financial incentives when they institute new vaccine programs with newly created vaccines. Rotavirus was the example used in the article. The African doctors argue that pushing newly created vaccines creates neglect of vaccination for basic diseases such as measles. I had been under the impression that the profit margin for newly created vaccinations was relatively small, i.e. not large enough to influence such funding allocations or disrupt existing vaccination programs. Your financially disinterested thoughts? Have you also been hearing developing world anecdotes about shortchanging older, well-proven vaccines in favor of flashier, costlier new ones?" Lark is a lay reader in Goodrich, Texas.
I don't really have any insights into this, though I must say that I've seen people quibble over pretty small amounts of money.
Well, the Rota vaccine was developed and delivered to Africa and other third world nations because that's where the deaths are from that disease.
Right.
Yeah. I think that's a case where it's a focus on what's the biggest problem.
Mm-hmm.
And yeah, measles is a problem, and you'd love to be able to vaccinate. You'd love for everybody in Africa to get the same series of vaccinations that people in Europe and the U.S. have access to, but it doesn't seem to be happening. So where do you start?
I heard a presentation by a WHO person recently on the measles eradication project. And, you know, there are big gaps in Russia, in the former Soviet states, and in Africa. And he said it's just a matter of getting people in and immunizing. And right now they're focusing on polio. They can't do everything. And their attention will slowly turn to measles, and they'll deal with that.
Which is ironic because one of the big selling points for the polio eradication campaign was that it would help build the infrastructure for measles eradication and other vaccination campaigns.
Mm. Yeah. I have not encountered this notion that a newer vaccine is displacing measles. I think Rota was pushed in because people were dying, kids were dying of dehydration, etc., and it worked. But they still want to do measles as well. So that's my 2 cents. But I don't have a lot of insight either, as Rich said.
Yeah, I don't know if there are financial incentives or disincentives for particular vaccines. And it may also just be a question of what's being donated right now.
Yeah.
Mm-hmm.
Alan.
Jim writes, NIH recently offered 2 presentations that your postdoc and younger listeners might appreciate. One deals with using LinkedIn effectively, 7th in the How-To series, while the other is about careers in science writing, 6th in the How-To series. Both sounded pretty useful. The video version of the LinkedIn talk includes visual demos of computer screens, so the video version of that one is more useful. Alan might like to comment on the science writer talk. Both talks are professionally oriented and detailed enough to be useful to job hunters, job suppliers, and anyone interested in improving communication skills. Science writer advocates might also appreciate this item I put in my blog. Professional listening Conversations Network seeks people to listen to podcasts and write summaries of them, for which it pays $5 each. The link will produce the application process, which consists of producing a short and long summary of a sample podcast with photo, biography of the speaker, and resources, exactly what's seen for each entry at this typical page that he links to. Once accepted, you have to produce 2 reviews under the eye of a mentor who will get your payments for that work, but payment will go to you starting with the 3rd product. You need a PayPal account where payments are made. It can take a month until you get your first payment, and they may be bundled rather than one at a time. You'll be paid to learn.
It's funny. Pay people to listen to a podcast. I can't imagine that they would be particularly interested if you have to pay them to listen.
Well, you're not paying them to listen. You're paying them to write up a summary.
They don't have to.
Ideally, as people are listening anyway, well, take the money to write up a summary.
That's the only way it would make sense, because $5 to write up a summary of a podcast that you weren't going to listen to is
not a lot, right?
Not a very good return on your.
I'm not doing that.
No, I'm definitely not doing that.
Boy, for a TWiV,
it'd take more than that for conference coverage.
Take an hour and a half to do a TWiV for $5 for an hour and a half. That's below minimum wage.
Yes,
way below.
Yeah,
and writing up a cogent summary of a long talk is harder than it looks.
Yep. All right. Rich is next.
Let's see. Luis writes, Dear TWiVers, once again, thank you very much for your excellent podcast and your service to the community of researchers and educators in the biology field, and specifically virology. It has been a while since my last email. Don't worry, you haven't lost a fan. I haven't missed a podcast since TWiV 113 when I learned about your show. Your recent podcast, TWiV 173 and 183, discussing about bats harboring influenza and paramyxovirus respectively, brought back to my attention a paper published last year in PLOS Pathogens, where, using deep sequencing techniques, the authors reported finding Ebola virus-like filovirus sequences in bats from caves located in Asturias, northern Spain. As a native of that part of the country, I was surprised and interested in the whole story. Of course, there was no evidence of infection or disease, and they were just only sequences. But anyway, I found it very interesting and intriguing. I was wondering if this kind of search for filovirus has been carried out in other countries outside Africa, maybe in the U.S. On a related issue, I recently learned about a huge virus hunting project known as Global Viral Forecasting Initiative. Do you have any information about it? Perhaps it would be nice to have one of their leaders talking about it on your show. Thank you very much for your efforts to promote science in general and virology in particular. Long life to TWiV. And Luis is from Spain, obviously, from the Centro de Biológica Molecular Severo Ochoa in Madrid.
We read a letter of his last week, too. I remember this filovirus discovery in Spain. I read that while I was in Brazil last year, and I thought we should talk about it, but we never did. They basically found some sequences in bats, but not exactly Ebola, but related. They're Ebola virus-like, maybe ancestral. Who knows? Whether people are looking elsewhere, I guess they are, but I don't know. I haven't heard of any results. I'm sure.
And this global viral forecasting initiative this is interesting. I was not aware of this.
This is a Nathan Wolf endeavor.
Uh-huh.
He's got money together to look for viruses.
Yeah. Google, the Wellcome Trust, and the Skoll Foundation.
Yeah.
And apparently he's there's a for-profit and a non-profit arm of this.
Very interesting
thing.
Hey, Nathan, you want to come on and talk about it?
Yeah.
All right. Let's see.
I think it's your turn.
Rich, me, yes. Jim writes, just in case you haven't seen this, sort of timely after your immunology podcast, perhaps your son, the game player, can evaluate. He sends a link to a game called Immune Defense. It's in beta. It's basically, I guess you run an immunology system and fight off invaders.
Immune Defense is always in beta.
Let's see. What do you think you're supposed to learn in this game? What do you want to learn? What is an LPS receptor? What is a protein? Please share your feedback with us. I could show this to my son, yeah.
Yeah, have him play it.
He'd probably just wave it off after 5 minutes, but I'll do that. And anyone else who's checked it out, let us know if it's any good. Thank you, Jim. Is it your turn now, Alan?
I think it is my turn now.
We're going counterclockwise.
You get the long one.
Right. Stephen writes, I thought you might... I'm probably going to summarize this.
He's got some good points, though.
He does.
Yeah, he does.
I thought you might like to look at Michael Spector's piece in the March 12th, 2012 issue of The New Yorker entitled The Deadliest Virus. It might seem out of date at this point, but it's featured on the New Yorker website's New Health section. This kind of article could get cited in classrooms, etc. The article's behind a paywall, but the issue's in libraries. I'm not qualified to do a point-by-point analysis, but I saw a number of errors, or at least questionable statements. Of course, some of these points could be called picky. I don't expect a response. I just wanted to bring this to your attention. You said you're not qualified to do a point-by-point analysis, and yet you actually have done a pretty good point-by-point analysis.
I would say yes, indeed.
Great job.
He points out, he marks this by page number in the, I guess it's the print edition. There's no mention that the 1918 flu was an avian flu. The opening discussion could be read as contrasting avian flu with the 1918 flu. The definition of pandemic, point 2 of this definition given in the article is it would have to kill them. It would have to kill humans. So I'm not clear on point 3. It would have to spread easily. How important is easy in defining spreading of a pandemic? So just discussing the meaning of the word. A description of the annual meeting of the European Scientific Working Group. Were the scientists astonished at Fouché's presentation? Had it been carried out in secret up to that point? Description of Fouché's work as, quote, simply transferring avian influenza from one ferret to another had made it highly contagious. My understanding from TWiV's discussion of the now published paper is that there was a lot of genetic work done on the virus, and the issue was whether the virus could spread from ferret to ferret at all. And that's correct. The article claims they had altered the genetic sequence of the virus in a variety of ways. That had no effect. And again, that actually did have an effect. A couple of other relatively minor, there were a couple of these that specifically I think deserved issues of number of segments in the genome and other little factual glitches.
This one, Fouché's achievement.
Yes. Fouché's achievement was a quote from the article, Fouché's achievement was to place all 5 mutations together in one virus, which meant that nature could do precisely what he had done in the lab. The first part of the sentence contradicts the first point I made on this page. The second part of the sentence, is that true? Can we assume that any lab-built genetic construct could arise naturally? Of course, in this case, the intent of the research was to investigate what genetic changes would be needed for transmission.
That's a good point, yeah.
Yeah, it's a very good point. Just because you can do it in the lab doesn't mean that you could have this arise naturally. And in the TWiV where we talked about this paper, that's actually a major issue. Is there some selective penalty against the virus when you make these mutations that's going to affect it out in nature that you didn't see in the lab? Let's see, Michael Osterholm is characterized as, quote, one of the nation's leading experts on influenza and bioterrorism, and is quoted extensively. He is a leading expert on bioterrorism, certainly, and in influenza as well, and is a major public health researcher. And he obviously provides lots of juicy quotes for an article like this. The article said regarding the 1970s swine flu, most virologists familiar with the outbreak are convinced that it came from a sample that was frozen in a lab and then released accidentally. I've never heard this. Is it true?
It's a speculation. No one's ever proved it. Osterholm uses it as an example of why scientists need to do things under great biohazard conditions, and it's just never been proven. It's a speculation. So it's really inappropriate that he uses that as an example, I think.
Yeah, this is. I am certainly not convinced that came from a sample from a lab. It could have.
Could have, yeah, but no one's proven it.
No. Let's see. The labs in Rotterdam and Wisconsin where the H5N1 ferret work was conducted were BSL-3 facilities. And my understanding from TWiV's discussion of the now published paper is that it was actually done in BSL-4. It was BSL?
I think it was BSL-3 plus.
Yes, that's correct. It's 3 plus, yeah.
And then... Fouchier's virus, which now sits in a vault within his securely guarded underground laboratory in Rotterdam, has fundamentally altered the scope of the biological sciences. Seems like hyperbole to me. I would agree.
But the next one is hyperbole, too.
From Osterholm, those researchers have all of our lives at the end of their fingers.
Oh, come on, please.
So
I like this. Worldwide pandemic is redundant.
It's true. He's right.
It's true.
And more in the same vein comes up elsewhere in the article. This is this is an article by somebody who was clearly very persuaded by the more inflammatory rhetoric on the topic.
Mm-hmm.
I like this one on page 5. It's not clear when or where the research will continue. If the New Yorker had delayed publication for a month or two, this would have become clear. It's really a lot of good points.
Yes.
Did a very good job debunking the article.
So we will be happy to publish this. And it's just, I mean, you get articles like this that are full of hyperbole and others that are more measured. And if it's in the New Yorker, people are going to read it and say, they must be right. It's the New Yorker.
The New Yorker is capable of both. Some of the articles seem to me spot on, and others are questionable.
Right. All right. Let's move on to the next one. Is Rich's turn.
Where am I? Hang on.
Okay.
Sijun. Greetings, TWiVers. I am Sijun Zhang, a graduate from Wisconsin-Madison, and waiting to enter grad school in the Harvard virology program.
Nice.
Good
job.
I am currently residing in Singapore, where the weather is 29°C and 84% humidity, but usually it is 33°C and a gazillion humidity.
That's what I've heard.
I have always been a great fan of viruses and have always been interested in using molecular virology as a tool to understand ourselves better. Interestingly, although I have yet to actually have a chance to work on or with viruses, I have been lucky enough to have a great education from established virologists like Paul Alquist, Ann Palmenberg, Rob Kelgetta, and Thomas German. Sadly, I never had the chance for any interactions with Yoshi Kawaoka. So that's all in the Madison virology program. It's a very strong program. Ever since discovering your podcast 2 years back while researching for virology programs for grad school, I am addicted. It is hands down the best podcast I know. It is a great source of information for anyone on viruses and always a great way to keep up to date with current information both within and without the virology world while learning about scientific writing, ethics, biosafety, etc. with a healthy dose of humor mixed in. Humor? I love leeching off all the great information from Vince, Rich, Alan, and Dick, although the latter may be more for anecdotes. Oh, I jest. TWiV is a great companion for me when I go on long runs, giving me a comprehensive workout both physically and mentally. I have always wanted to write to TWiV, but have yet to come across anything interesting to share or have anything to contribute. That changes today. Here's a great listener's pick for you guys. And I'll come... okay, she describes it. It is a little game called Plague Inc., which I came across and was hooked on. Of course, my first reaction was, I got to share it with TWiV. In the game, you play the role of viruses, bacteria, prions, etc., as you try to mutate your strain to infect and wipe out the whole world, starting from patient 0. Think of it as epidemiology for pathogens. I highly recommend trying this out on an iPad, although you can play it with the iPhone too. All in all, keep up the awesome work. I look forward to many more episodes of great fun and great learning from TWiV. Cheers, Sishun. There you go.
Cool.
I did not actually purchase this app. It's 99 cents. Being not much of a gamer myself, but I had a look at it on the iTunes store, and it looks pretty interesting. So
there are quite a few of these kind of infection games we've talked about before. The company that developed this one is called Endemic Creations.
Cute.
Very nice. All right, thank you. And,
yeah, that was a very laudatory letter. That was very nice. And good luck with our
Harvard
neurology program. That'll be just great.
All right, the next one is from Lance. Dear Vincent and team, I thoroughly enjoyed your podcast on reforming science. Very thought-provoking. There was, in my view, one thing missing from the discussion and from the articles, though it's of a slightly different nature and not such a systemic problem. That is, improving management and leadership in science. This is a problem in common with the only other field I have direct experience of, the field of clinical medicine. In both areas, at least until relatively recently and still persisting in many quarters, exists the dogma that being good at something also means you will be good at managing other people doing that same thing. This is most definitely not so in my experience, and I have seen some truly appalling efforts at trying to manage and motivate a group of people. This is something that most scientists have no formal training in, and nor, as far as I can tell, does it play much role in promotion or the awarding of funding. You can have the best ideas in the world, but if you can't work effectively with other people, they aren't much use. Paying more attention to the role of management and leadership in science would lead to greater productivity, in my view. Thanks again for a great show. It is so true.
Absolutely.
We don't get any management training.
None.
See Dilbert.
Yeah.
Yeah.
Yeah, this is a well-known phenomenon in all technically oriented fields, probably in all fields, that people get what was the Peter principle. People get promoted to their level of incompetence and stay there.
Yeah, that's right.
I wasn't even aware of the fact that management, in fact, is at least some people think it's a trainable discipline. A teachable discipline, till I had some interactions with business school fairly late in my career. And, I realized they taught courses in managing people and all of this kind of stuff. And I thought, holy cow, you mean you can actually learn how to do this? That would have been handy.
Yeah, there is a lot of it that can apparently be taught. You can improve somebody's management by sufficient training.
Yeah, so you learn how to do science, and then all of a sudden you're running your own lab, and you're hiring people, and you've got graduate students and that kind of stuff. And lo and behold, all these people come with personalities.
Yeah.
You bet. All right, let's do one more email, and I think it is Alan's turn. Is that correct? Yes, I
think it is.
All right.
Richard writes, hello, Vince and co-hosts. I'm never quite sure who will be on the podcast. You swipe things about quite often. That is likely a good thing. The weather here in Western Supermare, England, is that right? Western Supermare, or do they pronounce it mare just to annoy the somebody? Well, anyway, Western Supermare, England. It is cloudy and 11C, though it will get warmer and brighter later in the day. It's just a quick note. I'm sure you have already heard of the Genome Compiler project. I've just found out about it from TWiT. They have an interview with the creators, TWiT Live Special 126. I have yet to decide if I like the idea, but it seems like the beginning of something big. I guess time will tell. I can see that this is something that the human race needs, but I'm not sure if the potential to do bad things outweighs the benefit. I must get to work, unfortunately. However, I'll have the latest episode of TWiV to listen to while on the way. Thanks again for the podcast. Keep up the good work. And Richard sends a link to this genome compiler I didn't really look at this much, but I gather it's a front end for the genome databases.
So the idea is that, yeah, it's a front end, and people can work on modifying sequences and developing your own proteins, perhaps, and there's even a way to get it synthesized if you want. It sounds very strange to me. So this is basically a for-profit company that's trying to make money off of this. And you download a program, and then you mess with the genome. And the idea is to get everybody doing this and maybe come up with something interesting. And I was looking at the interview of this guy on TWiV, and he was saying, oh, yeah, you can make your own virus or bacteria, which, as we know, is just not possible. You can't make your own anything. So I don't really know what the output of this is. I think these guys are a little bit ambitious, and I'm not sure what they're trying to achieve.
Yeah, because I guess the idea is that this would appeal to garage biohackers or some other group that wouldn't already be familiar with processes for ordering oligos.
Yeah. I mean, basically you get a sequence, right? And then there's a nice interface for messing around with it, cutting and splicing.
Right.
And then you go make your oligo, and that's how they probably make money. And the guy said, you can get an oligo, or they'll even give you a plasmid with your sequence on it, and you can put it in cells and express it. It seems crazy to me.
It might have educational applications. I could picture using this maybe in a classroom and doing some kind of modification that would have a specific outcome and ordering up your plasmid, although that kind of takes a lot of the fun out of it in terms of teaching the material.
Yeah.
But there might be something like that. I don't know if this thing really has legs.
It just seemed not terribly useful. Of course, we're used to doing all this ourselves, so
yeah.
I guess we're just jaded, but I don't know if anyone's had any experience with it.
Well, it's currently, they say it's software is an alpha release, so it's not even at beta yet. But I don't know. Maybe it'll take off and they'll be the Google of the genome or something. But I don't know quite where that's headed.
I'm skeptical. That's my take.
Me too.
The article that he sends has several other sort of sequence mashing programs or products in it. Mm-hmm. It's going to be, as we accumulate more sequence, there's going to be a lot of sequence mashing products out there.
Sure.
And someone will rise to the surface.
Maybe.
I mean, if you're going to take sequences and hack them together and just make it, you're not going to get much. Unless,
there's too much we don't know.
You
can't just put it together and make your own book.
But I guess their idea is that if millions and millions of people do this, something good might come out of it. Who knows?
Well, some profit would certainly come out of it if you're getting commission on each sale.
I think that's what bothers me, is that they're going to get a part of each oligo sale. And, that's driving this. I don't know. Sorry to be negative, but I don't see the usefulness of it. All right, let's do some picks of the week.
Okay.
Alan, what do you have for us?
Let me scroll down past all these other accumulated emails.
Yeah, my goodness, there's just tons of these.
Sorry. Yeah, we'll get through them.
Yeah, eventually. And then more will have accumulated, and that's fine too. So my pick this week is something that a friend of mine, another science writer, Marcy Stone, just sent me today. This is a piece that appeared in Nature Reviews Microbiology. The August issue just came out a couple of days ago. It is the Microbial Olympics. And this is you can download it for free with a free registration on the site. It looks like a paywall when you get there, but you can sign up, register for free on the site, and download this article, which is a fun, humorous take. I gather Mary Yule is the instigator behind this, and they put together these different events where they describe how several microbes might do. So they have a sprint, they have a boxing match, and there's commentary on how the match is proceeding. They have the 100 micrometer freestyle swimming.
Yeah, good.
And that actually is the subject of the supplementary data. So you can watch a movie of the actual 100 micrometer freestyle swimming competition, and see the various bacteria swimming along and see who wins the race. I won't spoil it for you.
It's very cute. Nice.
It's a neat idea, and it gets across some good concepts in bacteriology and virology.
Excellent. That's cool.
Very nice. Rich.
This is something I learned about at the POX meeting. It's the PharmaJet needle-free injection system. You guys may be familiar with this, but I was not. This is a device for giving injections that does not involve needles. And the way it works is that there's a syringe, that is a cylinder with a plunger in it, that will hold up to a 0.5 ml that you can load with your vaccine or whatever off of a regular vaccine vial, and then you load it into this device, and there's a sort of a container for it that cocks it. And basically what it does is it sticks the syringe into this thing, and the plunger is cocked on a big spring. And what that does is push the plunger through the syringe and forces the fluid out through a hole that they say has the diameter of 3 human hairs at a very high velocity. So you just hold the end of the syringe up to somebody's arm and push the button, and the fluid comes out at such a narrow orifice at such a high velocity that without a needle it just penetrates the skin. And you can deliver up to a 0.5 ml. They say they've got an intramuscular. There's a YouTube video that shows how this works, and they've got some cutaways of some experiments done with dyes so that you can see how it compares to a needle injection, and it's basically indistinguishable. They've got intramuscular delivery. They've got one that's subcutaneous. I don't know how they would manage that, and they don't detail that, and they're developing one that's intradermal. They don't claim that it's pain-free, but apparently it is less painful than a needle, and it doesn't have a lot of the needle problems associated with it. So disposal, needle sticks, reuse of needles. These cartridges can't be reused. It's more expensive than a needle now, but when they get to high volume, they're hoping that it will be competitive. So I thought that was really interesting.
So what does this cost compared with a needle? Do you have any idea?
I don't know. I did know. I don't have it right at my fingertips. I saw it quoted somewhere, but I don't have it right now.
So this means you don't need a health care trained.
Yeah, that would be another
personnel.
Advantage. You ought to be able to use this. Anybody ought to be able to be trained to use this really quickly.
Yeah.
Oh.
Yeah, this is cool. This is, of course, part of a genre of new products and patents and such that have come out recently trying to do needle-free or different type of needle injection to get around all the problems with needles.
Yeah.
And this is just a really, really neat approach.
The guy who introduced it to me at the meeting said you could sneak up on a kid at night and vaccinate him, and he wouldn't even wake up. I don't know that that's true, but
Either you guys see this movie Prometheus?
No,
I have not seen that.
All right, so the reason I'm mentioning is this takes place way in the future, right? Mm-hmm. And there's a scene where they're giving someone an injection. There's this huge bloody needle they're using.
Actually, my wife pointed out to me that this PharmaJet thing is right out of Star Trek. You know that little gizmo that they hold up to them and it goes,
yeah,
and they've been injected.
But many futuristic movies still use big needles.
Yeah.
It's really funny because this is the future, right?
Right.
No more needles. Unless you need to do intravenous, I guess you'd need a needle still, right? Mm-hmm. All right. All right, thank you. My pick is a book just published by the American Society for Microbiology Press, ASM Press, called Microbes and Evolution, The World That Darwin Never Saw. It's a cool collection of essays edited by Roberto Coulter and Stanley Malloy, basically about evolutionary work using microbes, stuff that Darwin had no clue about. And it covers bacteria, fungi, viruses, all kinds of stuff. Very cool, like The Secret Social Lives of Microorganisms, Microbes and Microevolution, The Tangled Banks of Ants and Microbes, Symbiosis and Evolution. Really neat. Pretty nicely written chapters. And the cool thing is it's only $14.95 for paper or $9.95 for the Kindle edition. And it's a really nice book. So check that out. Really well done. And I just realized looking at this that, we have a TWiV bookstore where if you go and you buy a book, we get a penny per book or something. So if you want to buy something, just click on the link that we have at TWiV.tv. And now I realize we could put Kindle editions there as well. So I'll be putting both up because I have a feeling that a lot of people don't want to buy books anymore because I don't. And now we can do Kindles.
Yeah.
So do check that out. All right, we have a couple of picks of the week. We mentioned, I think, one or two already.
That were in the regular queue.
They're in the regular queue. There's one, I'll do two more. One from Ricardo, pick of the week, a TED Ed channel on YouTube. This may have been mentioned by someone before, but we'll bring it up. TED Education. It's always good to mention. And one from Christopher who writes, "Hello, TWiV guys. I just wanted to thank you for a great podcast and wanted to email a suggestion for a listener pick. Being a postdoc in molecular biology at the University of Michigan, I found another one of my favorite podcasts, Econ Talk with Russ Roberts, may be of interest to the TWiV audience. And it is an interview of Ed Yong on science, experimental replication, and journalism, which I found a thought-provoking discussion on many different topics, such as bias and experimental design." Wow, that's something we just talked about.
Yeah.
Yes.
Comes up all the time.
Good. By the way, I've noticed that since listening to TWiV, my incidence of viral illness has increased. Then again, it may be confirmation bias from my expanding knowledge that I have gained over the last 2 to 3 years, since I now know more about the nasty viruses out there. I particularly dislike enteroviruses after last year.
Medical student syndrome.
There you go.
Yeah.
In closing, keep up the great work, and it would be nice to see an Econ Talk/TWiV crossover in the future. I always learn a lot from both Econ Talk and TWiV, and I think you guys would have an interesting discussion. That's a good idea, right?
Sure.
Uh-huh. Yeah.
Do either of you know this this podcast?
No.
I don't.
You could talk about the economics of virology or something like
Yep.
that, right? Well, if he talked with Ed Yong on science, he could talk with us.
Sure.
That's a good
Sure.
idea.
Yep.
All right, so thanks, Christopher, for that. And that should do it. We have a whole list here of listener picks, but we'll save them.
Yeah.
We have these are all the emails, by the way, that I had in my queue. I spent like 2 hours yesterday pasting them into here. But then last night 4 more came in. It's great. Don't stop. We love them.
We could do sort of, this is virology and landers, right?
Yes.
It's incredible.
That could be a title, although probably would be violating some patent.
Probably.
No, I think that's fair use.
Yeah, I think so. Fair use. Did I tell you I did a video a couple of years ago at a Munich conference. I went to the party afterwards, and I took videos of virologists dancing, right? And I put it on YouTube because it's got a lot of well-known virologists there, and their labs like to watch them dance. Of course, there's music playing in the background, so YouTube flagged it as a copyright violation. If you film your wedding and they play some song, you can't put it on YouTube because it's a violation of copyright. It's not like you're making money.
Yeah.
It's weird. That'll do it for TWiV 192. We're going to be in Madison, Wisconsin when you hear this, or we will have been there by the time you listen, depending on your schedule. Rich and I will be doing a TWiV with a couple of the speakers from there, so that should be fun. Check it out.
Are you going to be on video?
No video, no live, just recorded audio. As we discussed earlier, ASV is not at the forefront of social media. If it were ASM, we'd be live streaming the video, sure. Right. TWiV.tv, iTunes, the Zune Marketplace. Do leave a comment over on iTunes if you're new. That really helps us a lot. And send us your questions and comments to TWiV at twiv.tv. Alan Dove is in Western Massachusetts, but he's also at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
Very good to have you again, as usual, and we hope that you won't miss us terribly next week.
I will miss you terribly next week.
But we will all be together for the 200th episode.
Yes, that'll be fun.
Which will take place in your home state.
Yes.
Do you mean that I am finally going to get to meet Dr. Alan Dove in the flesh?
You are, yeah.
Yes.
Wow.
In a bubble suit, too, right?
Yeah.
Yeah.
Can shake hands through a bubble suit. Far out.
One of our hosts at that episode, Paul Dupre, he's at Boston University. He will be in Madison for ASV, Rich, so you get to meet him, too.
Excellent. Good.
That'll be a lot of fun. That'll be the 200th episode in September. Rich Condit is at the University of Florida, Gainesville, where Satchels is back.
Satchels is back in business. Man, you're making me hungry just talking about it. I went there last Thursday. Maybe I'll go again tonight. I've been thinking about that.
No, you can't go. You can't go.
You look at my Facebook page. Have a look at my picture. It's a nice big Satchels. It's the pizza I had last Thursday.
Nice. Well, as I said, I'll have to make a trip for some reason and just to go to Satchels.
It's a destination, I tell you.
Well, while I'm there, we could do a podcast.
We could.
I'd take Alan with me, and we could all go to Satchels.
Sure.
That's a great idea.
All right.
It's all good.
I'm Vincent Racaniello. You can find me at virology.ws.
You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.